The convergence of microfluidics and 3D printing represents a rapidly evolving domain in advanced manufacturing and material science. Microfluidics, the control and manipulation of fluids at the microscale, has revolutionized how we handle chemicals, biological samples, and functional materials. 3D printing, or additive manufacturing, has similarly transformed fabrication by allowing layer-by-layer construction of objects with unprecedented geometric complexity and customization. The synergy of these two areas is leading to novel processes, devices, and materials that expand the capabilities of both fields. In essence, integrating microfluidic principles into 3D printing processes can enable finer control over the deposition of materials, allow on-demand mixing and functionalization, and facilitate the fabrication of structures that incorporate internal channels, sensors, and complex architectures. Such hybrid approaches promise innovations in fields such as wearable electronics, tissue engineering, microreactors, sensors, and advanced functional composites. Understanding the interplay between microfluidics and 3D printing is thus essential for scientists, engineers, and innovators aiming to push the frontiers of precision fabrication. The field of microfluidics emerged primarily from the demands of micrototal analysis systems, UTAS, and lab-on-a-chip devices in the late 20th century. By leveraging microscale channels and controlled fluid flow, researchers achieved remarkable reductions in reagent consumption and analysis time. In parallel, 3D printing technologies have evolved from simple prototyping tools to a broad spectrum of processes such as stereolithography, fused deposition modeling, direct ink writing, selective laser sintering, and inkjet printing. Initially applied in rapid prototyping, 3D printing gradually expanded into fields as diverse as aerospace, automotive, medical implants, and consumer products. The intersection of these technologies gained momentum as researchers recognized that microfluidic techniques could address some of the challenges in 3D printing, such as achieving high-resolution multi-material deposition or embedding intricate microstructures inside 3D printed parts. Conversely, 3D printing offered microfluidics a route to more complex, integrated, and three-dimensional architectures, something traditional planar microfabrication methods struggled to achieve. Microfluidics operates at length scales typically between 1 micrometer and 1 millimeter. At these scales, fluid behavior is dominated by laminar flow, surface tension, and diffusion-based mixing. The Reynolds numbers are usually very low, ensuring predictable fluid dynamics. Such precise control over fluid flow and droplet formation can be leveraged to achieve uniform deposition of inks, to encapsulate and distribute functional particles, or to create graded material compositions in three-dimensional structures. Many microfluidic devices create droplets of well-defined sizes and volumes. These droplets can serve as discrete packets of material feedstock in 3D printing, allowing fine-tuned control over deposition. By manipulating factors such as flow rates, channel geometry, and fluid properties, like viscosity and surface tension, one can generate droplets with a high degree of reproducibility. Droplet-based additive manufacturing can yield objects with intricately patterned internal features, tailored porosity, or spatial gradients of composition. Traditional 3D printing often relies on pre-formulated materials. Microfluidics, by contrast, can mix multiple components in real time during the printing process. This facilitates creating gradient materials, embedding active agents like cells or catalysts, or introducing nanoparticles for enhanced conductivity or mechanical strength. Through microfluidic networks integrated into the printhead, materials can be tuned at the point of deposition, opening doors to dynamic and adaptive fabrication strategies. Inkjet printing, a well-established technique in additive manufacturing, uses droplets on the order of picoliters to deposit ink. Integrating microfluidic principles into inkjet printheads allows for improved control over droplet size, uniformity, and frequency. Advanced nozzles equipped with microfluidic channels can handle complex fluids, such as suspensions of nanoparticles, biological cells, or high-viscosity polymers, and print them reliably. This combination also supports printing multi-material composites where different inks are routed through separate microchannels, merged, and then printed in precise patterns. Direct ink writing or extrusion-based printing relies on the continuous flow of a viscoelastic ink through a nozzle to build up structures layer by layer. Incorporating microfluidic elements within the nozzle enables in situ mixing, rheology control, and the formation of complex hierarchical materials. For example, researchers can co-flow multiple inks through a microfluidic nozzle, adjusting the flow rates to achieve gradients in composition. This is particularly useful in tissue engineering, where cellular bioinks must be precisely positioned to form biologically relevant three-dimensional structures. Stereolithography typically involves curing a photosensitive resin with a UV laser or projector. Integrating microfluidics into this process can improve resin handling, ensuring fresh resin is delivered to the build area with uniform properties, or enabling the localized mixing of functional monomers and additives. Microfluidic chips can be incorporated into the resin reservoir or the build platform, providing real-time adjustments to resin composition or photoinitiator concentration. Materials development enabled by microfluidic techniques. A significant portion of microfluidic-enabled three-dimensional printing focuses on polymers and hydrogels. Microfluidic controlled mixing allows for the fine-tuning of polymer molecular weight, cross-linking density, and additive incorporation. In bioprinting, microfluidics helps precisely position cells within hydrogel scaffolds, ensuring cell viability and spatial organization that mimics natural tissues. Controlled droplet generation can also produce microspheres or microgels that serve as building blocks for porous tissue-like constructs. Although more challenging, integrating metal or ceramic particles into a fluid medium for three-dimensional printing is possible with the help of microfluidic devices. By controlling suspension stability, particle concentration, and flow characteristics at the microscale, it becomes feasible to print objects with improved mechanical, thermal, or electrical properties. For instance, metal nanoparticle inks can be formulated in microfluidic reactors to maintain consistent concentration and size, and then printed to form conductive traces for electronic devices. Functional materials such as conductive polymers, stimuli-responsive hydrogels, magnetically active composites, or piezoelectric inks greatly benefit from microfluidic control. By adjusting the material parameters in real time, one can print structures that change shape, conductivity, or mechanical properties in response to external stimuli. Microfluidic mixing can also incorporate light-sensitive, pH-responsive, or thermal-responsive components that lend smart behavior to the final three-dimensional printed object. Device fabrication and microfluidic architectures. One of the most promising applications of combining microfluidics and three-dimensional printing is the direct fabrication of objects that contain embedded microfluidic networks. Instead of laborious multi-step lithography and bonding processes, a three-dimensional printer can directly fabricate channels, chambers, and reactors within a part. This enables fully integrated devices such as lab-on-a-chip systems, wearable biochemical sensors, microreactors for chemical synthesis, or advanced heat exchangers with optimized fluid paths. Three-dimensional printing's layer-by-layer approach can create intricate internal geometries not possible with traditional machining. Coupled with microfluidic design principles, printing can achieve channel dimensions and network complexity that rival the capabilities of planar microfluidics. For example, one can design fluidic architectures that split flows into hundreds of channels for combinatorial chemical screening, or create complex vascular networks within a biomedical scaffold to deliver nutrients and remove waste products. Beyond just fluidic channels, microfluidic-enabled three-dimensional printing can incorporate sensing elements, electrodes, and wiring. Conductive inks can be printed in carefully designed patterns to form integrated sensor arrays, signal lines, or heaters embedded directly into the device. This integration reduces assembly steps, improves reliability, and can create novel smart devices that monitor their own fluid flows, detect chemical species, or control reactions autonomously. Applications across disciplines. In bioprinting, microfluidic control is invaluable for managing the delicate conditions needed to keep cells viable. Microfluidic-assisted three-dimensional printers can deposit cell-laden hydrogels with precise spatial patterning. This is key to constructing complex, functional tissues like vascularized organoids, muscle fibers, or neural networks. Such engineered tissues have potential applications in drug testing, disease modeling, and ultimately in regenerative medicine. By printing soft materials with embedded fluidic channels, it becomes possible to create soft robotic components that move and adapt their shape when pressurized fluids are introduced. Microfluidics enables the optimization of these internal networks for highly responsive and energy-efficient actuation. These devices can serve as grippers, artificial muscles, or adaptive surfaces for advanced robotics applications. Three-dimensional printing with integrated microfluidics allows for the creation of portable, on-demand chemical reactors that can synthesize compounds under controlled microscale conditions. By tailoring the geometry of mixing channels, reaction chambers, and heat exchangers, these devices can optimize reaction kinetics, yield, and selectivity. Pharmaceutical and fine chemical industries stand to benefit from such customizable, continuous flow reaction systems. Microfluidic-enabled, three-dimensional printed devices can be employed in water purification, sensing of environmental pollutants, or optimizing energy storage systems. Microfluidics provides precise control over the structuring of electrode materials in batteries or fuel cells, improving efficiency and performance. Likewise, microfluidic chemical reactors can assist in producing advanced materials for solar cells or catalysts that degrade pollutants more effectively. Challenges and limitations. Not all materials ideal for three-dimensional printing are easily compatible with microfluidic handling. Some inks may be too viscous, prone to clogging, or may quickly cure and disrupt the fluidic control. Identifying or formulating materials suitable for both microfluidic manipulation and reliable three-dimensional printing remains a key challenge. While microfluidics can improve resolution, it may also limit throughput due to low flow rates and small nozzle diameters. Balancing high precision and complexity with manufacturing speed and scalability is an ongoing concern. Achieving industrial-scale production may require parallelization of microfluidic printheads and advanced flow control strategies. Incorporating microfluidics into established three-dimensional printers requires careful engineering of hardware and software. Custom printheads, fluid pumps, mixing chambers, and integrated sensing systems must be developed. Additionally, slicer software and CAD tools need to handle fluidic architectures and multi-material instructions, adding complexity to the design-to-print workflow. Some three-dimensional printed parts with embedded microfluidics may require additional steps like curing, cross-linking, or sintering, and ensuring channel integrity after these steps can be challenging. Maintaining consistent quality and reducing defects like channel collapse or contamination is crucial for reliable device performance. Future perspectives and research directions. Continued research in materials science is needed to develop inks that are stable, biocompatible, tunable, and functional under microfluidic conditions. This includes conductive polymers, stimuli-responsive materials, and bioinks optimized for living cells. The ability to print next-generation composites, like those including two-dimensional materials such as graphene or perovskites, could unlock new applications in electronics, sensing, and photonics. Advanced control algorithms and machine learning techniques hold promise for optimizing microfluidic-driven three-dimensional printing. By analyzing data from sensors embedded in the printer, these algorithms can adjust flow rates, curing times, and print paths in real time. This closed-loop feedback control can dramatically improve print quality and material properties. As the field matures, we may see open-source platforms that integrate microfluidic modules into widely available three-dimensional printers. Modular systems could allow researchers, educators, and small businesses to experiment with microfluidic-enabled printing without investing in custom hardware. This democratization of technology could accelerate innovation and bring new communities into the field. Microfluidics will increasingly be combined with other advanced manufacturing techniques. Hybrid approaches that integrate lithography, laser sintering, or robotic assembly with microfluidic dispensing might yield multifunctional devices with unprecedented complexity. Cross-pollination with fields like synthetic biology, artificial intelligence, and Internet of Things will likely lead to transformative applications not yet imagined. Conclusion. The integration of microfluidics into the realm of three-dimensional printing is ushering in a new era of precision fabrication, functional materials, and device innovation. By exploiting microscale fluid control, researchers can create complex architectures with embedded channels, gradient materials, and dynamically tuned properties. These capabilities enable the fabrication of next-generation biomedical devices, advanced sensors, responsive systems, and specialized chemical reactors. While many challenges remain, such as material compatibility, scaling up production, and integrating seamlessly with existing platforms, the progress made over the last decade has been remarkable. With continued interdisciplinary collaboration between engineers, chemists, biologists, and material scientists, the synergy between microfluidics and three-dimensional printing will no doubt yield groundbreaking discoveries and technologies. As these methods mature, we can expect to see increasingly sophisticated devices with integrated microfluidic functionalities, fueling advances across a wide array of scientific and industrial sectors. In short, microfluidics for three-dimensional printing materials. Devices and technology is not merely an intersection of two fields. It represents a powerful union that could redefine how we approach fabrication design and innovation. By mastering the interplay of liquids at the microscale and structures at the macroscale, we stand on the cusp of a new landscape in additive manufacturing, one where materials are programmed at the point of deposition and where complexity and functionality are limited only by our imagination and creativity.